All of this research is currently preclinical, and so not exploring any agents that are clinically available for patients, but starting to be able to assemble these into kind of a foundation to really be able to build from in the future. So we're excited to join for another video here talking about research in chromophobe kidney cancer. And we're excited to have Samer here to share with us about his latest paper targeting FSP1 in chromophobe kidney cancer. So before we dive in, can you share with us a brief introduction about yourself and what drew you to studying chromophobe kidney cancer?
Yeah, absolutely. Thank you so much, Katie, for having me. So my name is Samer Salem, and I graduated from medical school maybe a little bit over 3 years ago. And after graduating, I joined Dr. Elizabeth Henske's lab at the Brigham and Women's Hospital in Boston. And I've been working on chromophobes since. And it's a fascinating cancer, honestly. The biology is so intricate, so interesting. It's such in a lot of ways, it's a unique cancer. It has these, a lot of things that make it unique, such as the abnormal mitochondria that we see in it, and how these mitochondria accumulate to a point that makes the cell have really a distinct appearance under the microscope. But also because this cancer, because of its rarity, but also because of its unique and interesting biology, has not been studied a lot. And so we started working on it, and we are trying to build momentum to hopefully move what we did in the lab to the clinic and to the patients so that we can see a real world impact of what we did.
Awesome. Yeah, that's probably been one of the most exciting things about seeing the research that's been coming out of Dr. Henske's lab over the past few years, is being able to see all the progress. I think there was 3 papers just in the last few months that have come out. So we're so grateful for the work that you all are doing. And I know ferroptosis has been a major area of interest and area that the lab has really been pursuing. Can you explain to us what ferroptosis is and how your work highlighting FSP1, what kind of mechanisms or what relation to ferroptosis it has?
Yeah, absolutely. So ferroptosis is a kind of distinct and unique or different type of cell death. It's, for example, different from other types of cell death. For example, apoptosis or necroptosis or pyroptosis. And if you look at the name ferroptosis, it has maybe 2 components: ferro, which comes from ferrous or iron, and ptosis, which is death. And so really from the name, you can see that it's an iron-mediated type of cell death. And what happens is that because the cells have iron and have oxygen, the iron and the oxygen are very active together, and they cause sort of a rust from within type of death. And the reaction that iron and the reactive oxygen species lead to basically drives damage to the fats that are essential components of, for example, the membranes of the cells. And because oxygen and iron are both predominant, are present everywhere in biology for billions of years, maybe 2.5 billions of years, since oxygen has been present in the atmosphere, cells have had to develop kind of multiple defense systems to basically keep ferroptosis at bay and to basically prevent it from happening kind of spontaneously. And the most studied way that cells sort of protect themselves against ferroptosis is through the glutathione and GPX4 system. And GPX4 is an enzyme which stands for glutathione peroxidase 4. It just uses glutathione to basically quench reactive oxygen species and to keep ferroptosis at bay and to basically prevent damage to fats. And then we also have another newer component that is more recently emerging, which is FSP1, which we talked about in the paper. And it basically stands for ferroptosis suppressor protein 1. And instead of acting on glutathione like GPX4 does, FSP1 acts on CoQ10 or ubiquinol and vitamin K to help them neutralize lipid damage.
I think that's really exciting that you talk about kind of that ferroptosis has multiple kind of mechanisms at play. We recently recorded a video talking about apoptosis in Chromophobe and some of the work that came out of Lisa Henske's lab on BCL-XL and showing that there's multiple mechanisms when that are like our cells in our body have to try to protect cells, which is really helpful for our normal cells in our body. But then it's learning how to target those when it's a tumor or cancer cell. It's really exciting to see how the basic science of understanding how these mechanisms work can help inform how you kind of turn all of those switches and make sure that you're only killing tumor cells eventually. So I really appreciate the fact that this is finding kind of another one of those mechanisms for ferroptosis. So what made you look at FSP1 specifically when it came to ferroptosis? It sounds like there was one kind of area and pathway that was already had been explored by the group. What made you guys decide to focus on FSP1?
Yeah, absolutely. So yeah, as you said, I mean, the field of ferroptosis has only been around for maybe 13 years, since 2012. And we first, the whole field started first by looking at glutathione and GPX4. And the whole FSP1 thing has only been emerging more recently and has only been identified in the last few years. And so we were looking at different ways to induce ferroptosis in chromophobia and cell carcinoma. And when we were looking at basically a data repository that we call the Cancer Genome Atlas, or TCGA, we saw that in chromophobia and cell carcinoma, you have extremely high FSP1 levels. Actually, out of all the type of cancers in the TCGA, chromophobia and cell carcinoma had the second highest increase in FSP1 compared to its own normal organ, which is in that case the kidney. And so when we saw that, we thought that this is interesting and we should look more into it. And we did look more, and within that same data set, the TCGA or the Cancer Genome Atlas, we tried to see how FSP1 levels or expression correlates with survival of patients with different FSP1 levels. And we saw that the higher the FSP1 level, the worse patient tended to do. They tended to live a shorter amount of time when they had chromophobia and cell carcinoma. And this also further strengthened our hypothesis that FSP1 is playing a certain role in chromophobia and cell carcinoma. And so we also looked at other functional data sets. And there we saw that higher FSP1 expression led to more resistance to GPX4 or SLC7A11 inhibition. And so as we started putting all these blocks together, it started to look like chromophobia and cell carcinoma might have sort of a unique dependency on FSP1 in order to be able to survive. And exactly like you said before, it's always this dependency or this vulnerability that we're looking at, because we want to find something that we can target. And if we target it, it will cause death to the tumor cells without causing death to our normal cells, and without causing toxicity to the patient. And that's where things started.
Yeah, so I think that's what I found the most interesting about this work and most promising, because I know kind of early exploration of ferroptosis, not just in chromophobe kidney cancer, but other cancers that are also susceptible to ferroptosis, was that in order to induce ferroptosis in those types of cancers, the treatments often are very toxic if you're kind of just targeting that one pathway. And so what I found really exciting about your work, we were talking earlier before we joined the call of how much difficulty I found early on in my diagnosis to read all of the charts. But I know that there's a chart that I've seen regarding your research that highlights kind of the dose of kind of the I don't know if it's IKE. Definitely correct me
Yep.
on which inhibitor it is, but combining the traditional approach to ferroptosis that has been explored in previous papers, along with targets that you've had for FSP1, and that you can kind of lower the doses by targeting kind of two mechanisms at once. Hopefully, that in the future could lead to kind of more accurately targeting these cancer cells while reducing the toxicity that for all of the other organs in your body. Can you speak a little bit more to that and kind of the approach or what led you all to exploring that approach and the importance of combining this research and how it kind of builds on each other over time?
Yeah, absolutely. So as you started by saying, yes, GPX4, SLC7A11 have had problems both in their toxicity. So we know that they can cause lower blood counts, be it red blood cells or white blood cells or platelets, but they're also toxic to neurons. And on top of that, pharma companies always had problems in trying to come up with drugs or basically chemical formulas that would be easily dissolvable and easily available in the body once we injected these drugs into the body, or once we administered them orally in preclinical models. And so that was kind of a problem. And so our approach was, if we only used a small amount of the GPX4 or SLC7A11 inhibitors, or maybe what we started with was genetic inhibition of GPX4 or SLC7A11. And when we inhibited these alone, cells tended to pretty quickly be able to resist that because they would be able to develop resistance by changing the by changing the expression of other genes. And so, but when we did inhibit GPX4 or SLC7A11, and at the same time inhibit FSP1, we saw complete cell death or almost complete cell death in preclinical models on cells in the lab. Whereas inhibiting each one alone, so inhibiting FSP1 alone or GPX4 alone, using genetic knockdown or genetic inhibition was not that effective alone. And so for us, being able to inhibit both arms at lower levels and seeing complete cell death is a great result, because then we're able to minimize toxicity while maximizing efficacy.
Yeah, I think that's what is so exciting to watch is this. And it, to me, as somebody who didn't understand basic science and translational science before and kind of really only understood the clinical side of it, when you see the clinical trials, when it's moved into drug development, I think that's been one of the most exciting parts for me to learn about the importance of basic science is because you're understanding those building blocks and then figuring out how to assemble them to create the end products that come to the clinical trials and really bring new treatments to patients. And so that shows exactly that process. It's really exciting to watch as somebody who about 5 years ago was diagnosed and saw how limited research there was in chromophobe kidney cancer. I can't say how meaningful it is to watch you all build those blocks to be able to kind of move that progress forward. And so one thing that I wanted to highlight a little bit that you touched on is you talked about kind of there's multiple approaches that you use to test this. So you had a hypothesis, you started to see it proven out in the data, and that there are multiple approaches you use to test that to see if kind of what you're seeing is validated. And one of them you mentioned is you used a knockdown approach. And can you explain what a knockdown is? This is something that I previously didn't understand but saw everywhere in papers. So can you explain what a knockdown kind of model is and what you can learn from those?
Yeah, so a knockdown is basically when we insert a piece of genetic material, and this piece of genetic material is oftentimes complementary to our gene of interest that we're trying to inhibit. And so it'll bind to it and basically inhibit its its transcription from just DNA to the protein, which is basically what is functional, because you need your gene to be made into a protein for it to be functional. And so when we talk about genetic knockdown, we're talking about pretty much inhibiting this transition from gene to protein so that you don't have the protein and are able to basically inhibit the gene without even having to target the protein specifically via a drug, just with this fragment of complementary genetic material. Very clean way of targeting only one gene, whereas oftentimes this is a little bit harder to do with drugs because they can have multiple they can target multiple proteins, or oftentimes the bioavailability of the drugs might affect things a little. So genetic knockdown is just a cleaner way to do it, especially at first, just to test the waters, kind of, just to see if your hypothesis holds up, and then you can move on into drugs.
Yeah, I think that makes a lot of sense when you think about kind of what you talked about earlier, that our bodies are kind of these like really complex like chemistry sets is kind of what I think of a lot of times. And so if you're using drugs to inhibit something, the body's probably going to try to express other things to try to compensate and bring those mechanisms into place. And so the description that you have for kind of targeting just that one singular protein expression helps kind of isolate to just the function of that protein. And I always find these, please correct me if I'm wrong, but is it the Western blot that usually kind of shows if this is how well you've done at blocking that protein?
Exactly, yes.
Okay.
Because on a Western blot, you stop seeing the protein on your blood, on your gel, and so whenever you stop seeing the protein, means that your gene has been inhibited. And I forgot to mention earlier, but it doesn't have to be only a complementary piece of genetic material. Sometimes you can edit the whole gene using what we call CRISPR, and that's another type of genetic knockdown. It's a knockout, oftentimes called, instead of a knockdown. But yeah, both are types of genetic inhibition instead of protein inhibition.
Super exciting. So I'll put an example of what these look like because I know when I first was learning this and I would see knockdown, and I would see knockout. I didn't understand any of it, but these little charts looked like little caterpillars to me.
Yeah.
So now when I see them, understanding what they mean is exciting. So I'll put those up there because I do think that they can help those that are kind of just learning or patients who might be interested in the science to be able to follow. And so those are usually where as I'm reading through a paper now, I can see, okay, so we can see that's the effect. Now let's move on and see what happened to that. So, all right, so we talked about how you use that to kind of validate FSP1 was really an area of interest to target and see how its effects could be impacted on chromophobe. You share in the paper that there were a few agents that you used to test targeting this. Can you talk a little bit about what those agents were and kind of what the next steps are, or if any of these are currently clinically available?
Yeah, absolutely. So the field of FSP1 has actually quite a few different inhibitors of FSP1 that are available. The most commonly used ones are ICFSP1 and FSN1. And we think that both of them do a pretty good job of inhibiting FSP1, including in vitro and in preclinical models, so in mice and so on. And we were excited about trying them, basically combining lower doses of these FSP1 inhibitors, be it ICFSP1 or FSN1, and combining them with inhibitors of the glutathione pathways, so inhibitors of GPX4, such as RSL3, or inhibitors of SLC7A11, such as IKE. And we again saw strong synergy when these two arms were used together or were inhibited together, even at lower doses, where we did not see significant cell death with only one of the two arms. When we combined the two arms, we saw almost complete cell death. And this synergy was very exciting for us because, again, as we said earlier, it would help us minimize toxicities while maximizing efficacy and really trying to get into that therapeutic window where we are really treating the cancer, killing the cancer cells without causing any or as little as possible toxicities.
Yeah, and that's really exciting, obviously, because that moves into being able to find new treatments for chromophobe kidney cancer. All of this research is currently preclinical, and so not exploring any agents that are clinically available for patients. But are any of these currently being explored, or what are the next steps to exploring moving some of these agents into clinical trials? That it takes quite a while usually to kind of move along, but what areas are you watching for to see if any of these agents might move closer to the clinic?
Yeah, so we saw that FSP1 inhibition might be effective in chromophobe renal cell carcinoma, and that was one of the first cancers where FSP1 inhibition was shown to be effective. And I think the momentum is slowly building in that area. And I think we need more studies on FSP1 inhibition in cancer specifically to basically build a strong case in order to move this development from the lab into pharma companies and then eventually into clinical trials and be able to offer it to patients. And so there's quite a long way to go, but data is starting to build, and we're very hopeful that we're gonna be able to make a difference for patients using this approach.
Yeah, so I think if we kind of go back to our analogy of those building blocks that we talked about, it sounds like before there were a couple of building blocks that had been started targeting GPX4 and targeting SLC7A11. And now we have kind of more building blocks that we have to build on with FSP1, but these are kind of newly discovered. So there weren't as many of these, but starting to be able to assemble these into kind of a foundation to really be able to build from in the future. So not available for any clinical trials or anything yet currently, but really helps understand how all of the pieces connect so we can set that foundation and work towards treatments that are more specific and might be able to be more tailored to the biology of chromophobe kidney cancer.
Exactly. And I think there's also more and more work being done that is adjacent to ferroptosis and FSP1 that can also help move the field forward. There might be efforts there are efforts that are trying to combine both ferroptosis and other types of cell death, such as apoptosis, but we're also trying to understand lipids in a better way because ferroptosis mainly affects lipids. And it basically denatures them so that the cell is not functional anymore. And there's a lot of research currently being done on lipids and what makes a certain lipid sensitive to ferroptosis. And as we start building more and more and more blocks, hopefully then something can emerge out of the field that is really strong and really effective.
Yeah, that's really exciting to know that there's progress and know things are moving forward and understanding the biology for chromophobe kidney cancer. So as we kind of close out here, the last question I have for you is what brings you the most hope for patients with chromophobe kidney cancer or the progress in understanding the biology of the disease to bring more effective treatments to it?
I think what really brings the most hope in the chromophobe sphere is really how much progress we've seen in understanding chromophobia and cell carcinoma in the last 5 or 10 years alone. It's been tremendous. Whereas in, maybe 5 years or 10 years ago, everything that we knew about chromophobia and cell carcinoma and all the drugs that we used to think might be effective for chromophobia and cell carcinoma and that we used in the clinic were actually just derived from its more predominant or more common cousin, clear cell renal cell carcinoma. And whatever worked in clear cell renal cell carcinoma, we just tended to use it in chromophobia renal cell carcinoma. And we now know that this is not the most effective approach for treating patients with chromophobia renal cell carcinoma. And it's really great to see all these mechanistic studies that are targeting Chromophobe in cell carcinoma, because when we understand the cancer better, we're able then to find ways to target it that are specific to this cancer and bring that increased efficacy to the patients. And I think this is the most exciting thing that I've seen. A lot more needs to be done. And I think we need to combine the efforts of both scientists and people who work in labs with the efforts of clinicians and clinical trialists and the efforts of pharma company, but also very importantly, bringing also combining our efforts together with patient advocates like you who are able to basically raise the profile of chromophobia in cell carcinoma, make the public aware that funding is urgently needed in this sphere to be able to catalyze more research and help us better understand this cancer. And I think over the past years, we've seen this come together, and this is really exciting.
I couldn't have said it better myself. That's what brings me the most hope for chromophobe kidney cancer. Like I said, being a patient who learned about chromophobe kidney cancer not even 5 years ago and knowing how little research was available then, and how much better we are starting to understand the biology of disease. And while we don't have new treatments that are available yet, although there is a clinical trial open for c-Kit, which targeting KIT was a paper that came out of Lisa Henske's lab recently as well too. So it's really exciting to know that we are right on the cusp of that. And that came from so many different mechanisms, including this kind of increased advocacy effort, but also that wouldn't have been able to be amplified if there wasn't funding for the labs through some of the larger grants as well too. So I'm very excited for the exact same reasons, and just want to thank you for the efforts that you've had in helping move this research forward. I'm really excited to watch these blocks continue to assemble and bring better outcomes to patients. So thank you so much for joining us and for sharing your paper and bringing more research to chromophobe kidney cancer.
Absolutely. Thank you so much, Katie. And thanks for everything that you do for the field.